This is the list of our group members that include myself, Alia Maisarah binti Azizan, Muhammad Izzuddin bin Jalaluddin, Nick Nur Azimah binti Azman, Nur Natasha binti Nazihan, and Rabiatul Adawiyah Nawarah binti Zulkarnain.
As for the introduction, phosphorylation is a process of synthesizing ATP, adenosine triphosphate, from ADP, adenosine diphosphate, MP, inorganic phosphate. It is a biochemical process in which a phosphate group is added to a molecule, typically a protein, lipid, or sugar. This addition of a phosphate group can profoundly alter the structure, functions, and interactions of the molecule, thereby regulating various cellular processes in order to guarantee normal function and balance of different components and structures of a living organism in terms of cellular health, resilience, and survival.
There are two kinds of phosphorylation, which are substrate-level phosphorylation and chemiosmotic phosphorylation. Substrate-level phosphorylation is the direct formation of ATP or GTP by transferring a phosphate group from a high-energy compound to an ADP or GDP molecule. Substrate-level phosphorylation occurs in the cytoplasm of cell, glycolysis, and in the mitochondria, Krebs cycle. Chemiosmotic phosphorylation is the third pathway that produces ATP from inorganic phosphate and an ADP molecule. Chemiosmotic phosphorylation occurs in both mitochondria called oxidative phosphorylation during cellular respiration and in the chloroplast called phosphorylation during photosynthesis. For the photophosphorylation, the chloroplast produces ATP by chemiosmotic phosphorylation. The energy needed to produce ATP comes from the sunlight during photosynthesis. And for the oxidative phosphorylation, the mitochondria produce ATP from chemiosmotic phosphorylation. The energy needed to initially pump H plus ion into the intermembrane space comes from glucose. The entire process is called cellular respiration.
Here are some key functions of phosphorylation. First, activating or deactivating enzymes. Many enzymes require phosphorylation to become active and perform their designated tasks within a cell. This allows for precise control over metabolic pathways and cellular processes. For example, enzymes involved in glucose breakdown for energy production might be activated by phosphorylation in response to low energy levels. Second, to regulate protein-protein interactions. Phosphorylation can alter the electrical charge and shape of a protein, affecting how it interacts with other proteins. This allows for the formation or disruption of protein complexes, influencing various cellular functions like signaling pathways, gene expression, and protein degradation. Third, to control protein localization. The addition of a phosphate group can act as a signal that affects a protein to a specific location within the cell. This targeted movement allows proteins to be present where they are needed most, ensuring efficient and localized cellular function. The fourth one, to alter the protein stability. Phosphorylation can influence how long a protein functional by affecting its stability. In some cases, phosphorylation might mark a protein for degradation, ensuring proper protein turnover within the cell. And the final one is to mediating cellular signaling. Phosphorylation plays a critical role in a signal transduction pathways. Extracellular signals can trigger phosphorylation cascades, where one protein phosphorylates another, and so on, amplifying the signal and leading to a coordinated cellular response. Overall, phosphorylation provides a versatile and dynamic way for cells to regulate protein function and orchestrate a multitude of cellular processes.
Next, we move to the process of phosphorylation. For the first one is a high-energy molecule substrate is used to transform a phosphate group to ADP to form ATP. The energy release will be used to bond the phosphate group to ADP, forming ATP. Next step is an enzyme is needed because they catalyze the transfer of phosphate group from ATP to specific target proteins. Next, a kinase enzyme recognizes and binds to a specific target protein substrate. The substrate protein has a specific amino acid residues that can be phosphorylated. And the last one is ATP is produced. The substrate protein is now phosphorylated. The addition of the phosphate group changed the structure and chemical properties of the protein, altering its function and interaction with other molecules within the cells.
In conclusion, substrate-level phosphorylation is a direct enzymatic process that produces ATP in pathways such as glycolysis and the citric acid cycle. It serves as a rapid energy source, especially in anaerobic conditions, demonstrating its crucial role in cellular energy production. Understanding this mechanism is fundamental to comprehending how cells generate energy efficiently across different metabolic conditions.